Okay, so for the discussion for this unit, I ask a couple of questions regarding the endosymbiotic theory, which has been developing over the course of the last decade or so in our understanding of how the first eukaryotic cell formed. And it's very interesting, and at least in the realm of biology, I'm definitely more of a symbiotic driven kind of biologist. Usually, I start off with discussing the human microbiome, and inside our digestive tract, as well as on the surface of our skin and other places, we have very characteristic bacteria, and it's really been demonstrated this day and age that most of us have a very unhealthy microbiome. There was a recent study that was conducted with primates in the wild. I don't remember which monkey species they were, but they're two separate species that were actually analyzed at the gut flora of captive monkeys and monkeys in the wild, and they found the captive monkeys, very interesting enough, were very similar to the human microbiome, the bacterial flora that make up our gut flora, anyways, were very characteristically in line with the captive monkeys. The interesting thing is the captive monkeys had a lot of the unhealthy diseases that are associated with our diet, and such as heart disease and various digestive ailments, that kind of thing. But with the primates that lived in the wild, they were much leaner, healthier, no obesity, diabetes, that kind of thing. And so it's suggested that the human microbiome is somewhat off. And we've estimated somewhere around 1,000 different species that live in conjunction with us. And it's probably even more. That's why I take I take probiotics, and I don't go cheap on it, is what I'm trying to get at. I think it's very important to pay attention to your microflora. And that's one example of a multiple symbiotic relationships occur, and that's kind of the norm. Well, this endosymbiotic theory, there's evidence has been discovered that suggests that the mitochondria were once free-living aerobic bacteria. The mitochondria today are most closely related to alpha proteobacteria, and they exist in our cells today. So way back 1.5 billion, so give or take several million years, it's thought that this alpha proteotype bacteria, aerobic bacteria, used oxygen as its final acceptor of electrons as it breaks down organic molecules to derive energy, uses oxygen. And that was sort of an odd thing back then anyways. Oxygen was being released by cyanobacteria in the environment, and so it was changing the conditions. And so this new relationship that began to become established, because this primitive host cell that took in this bacteria was thought to have been an archaea-type bacterial cell. And so by a process known as endocytosis, it took in this alpha proteobacteria probably numerous times. This probably played over tens of thousands, if not hundreds of thousands of years to establish. But a relationship established between this host archaea-type cell and alpha proteobacteria, or a aerobic type of bacterium, to where it was a permanent relationship between those two cells, one living inside the other cell, basically. And over time, this became a permanent relationship, an obligate relationship. And it's thought that that's where the mitochondria came from, as well as the chloroplasts and lines of algae and plants, the cyanobacteria becoming ingested in a host cell, the archaea type cell, and eventually became established as chloroplasts. Now, the fact that the mitochondria, an alpha proteotype bacteria, was inside the cell was probably enough to drive the formation of a nucleus. See, these archaea type cells, it's thought that they were heterotrophic and engulfing other cells to eat them, like an amoeba. And so to have an oxygen using cell inside your cell might be detrimental to the DNA. And because these cell types, the archaea host cell, would invaginate its plasma membrane to take things in to eat, it's thought that it may have haphazardly, one cell, two cells, whatever, however many, haphazardly surrounded the DNA, basically by accident, but it served as a protectant for that cell that harbored aerobic bacteria. And so it was probably something that drove the formation of a primitive nucleus, just the membrane surrounding the DNA at first, eventually becoming something that was selected for through natural selection. But that's kind of what the endosymbiotic theory is all about. And there's plenty of evidence out there that supports mitochondria come from bacteria and chloroplasts come from cyanobacteria. They both are about the same size. Both mitochondria and chloroplasts have their own DNA that's very similar to certain strains of bacteria. They have their own ribosomes. So mitochondria have their own ribosomes. Chloroplasts have their own ribosomes. Chloroplasts also have an interthylakoid membrane very similar to what's observed in free-living cyanobacteria. They also divide inside the cell by a process known as binary fission in the outside world with other cells, but they do a thing that's very similar inside our cells that's like binary fission. So there's enough lines of evidence out there that seem to really support the idea that a host archetype cell took in bacteria, aerobic bacteria, that eventually became established as mitochondria. And of course the other line that would run down the line of eukaryotic algae and plants also acquiring a cyanobacterial partner that eventually gave rise to chloroplasts. So hopefully that helps to clear it up a little bit. I know there was some confusion on the discussion board, but I thought maybe you'd want to hear it directly from my mouth. But if you have any questions, I would just highly recommend that you message me within the course, and I will respond accordingly. Thanks a lot. See ya.